going to be a little bit different than what's been done so far. We're not an antibody developing or researching company, but we actually provide solutions, hopefully, for people out there in the field actually doing the research and development on, for example, antibodies. So my name indeed is Lorenzo Fanchi. As said, I'm a Bioinformatics Project Manager at ENPICOM. And so yeah, today I'd like to talk to you about discovery and prioritization of novel antibodies using repertoire sequencing and the IGX platform. Let's see if this works. Yes, okay, perfect. So ENPICOM is a young health tech company. We are comprised of a team of experts, both in the fields of immunology, but also in bioinformatics and hardcore software development. And we have our purpose basically is to generate insights from immunomics data and ultimately, of course, to improve patient health care and improve the lives of patients. So we provide support in every stage of development, going from R&D in, for example, antibody discovery, but also TCR therapeutics, preclinical development phases, but also providing support in data analysis, for example, in longitudinal analysis in clinical trials. So finally, our main goal is to bring together the knowledge that is required to provide solutions for the complex problems that we face as immunologists in the field of immunomics. So just a little bit of background. I'm sure I'm going to be telling you everything that you already know, but the kind of data that we work with is B and T cell receptor sequences. So as you're likely aware, these sequences are formed after somatic recombination of various gene segments to yield alpha and beta chains in the case of TCRs or heavy and light chains in terms of antibody or B cell receptors. Due to the random nature of this recombination, immune receptors have exhibit a staggering diversity, allowing them to bind a variety of different epitopes through the CDR3 region of the receptor. Now, recent advances in next-generation sequencing technology now allow researchers to probe these receptor repertoires to unprecedented depth, and especially in the field of antibody development in particular, we think that NGS has the potential to be a transformative technology. And to illustrate that, or to give a number of examples of how we think this could work, I would like to show you some examples of the application of NGS in some of these discovery fields. Yes, so one interesting example here is phage display technology. So as you're most likely aware, many of you will have used or use phage display in antibody discovery. And phage display technology has traditionally been a very laborious and time-consuming process. So how it usually works, I think this should work, yes. So usually these biopanning procedures work by doing subsequent rounds of phage infection, washing, and for example, performing ELISAs to see if binding affinity is increasing overall. But there are some inherent limitations to the use of this technology, especially Sanger sequencing. So one of the issues that usually plague these kind of technologies is the fact that dominant clones can often mask potentially interesting sequences that are not present in such high abundances. And on top of that, actually, Sanger provides a very limited level of detection, so you're very likely to miss subdominant clones, which might actually have interesting properties, better binding affinities, or different sequence liabilities. Oh, this doesn't work. Let's do it here. Yes. So using NGS, we actually can sequence libraries after each panning round. So instead of using ELISAs here, we actually sequence what is left after this part of the biopanning round. And we can screen these in a very fast and cost-effective manner. So by doing this, we can track enrichment and impoverishment of either individual sequences or clusters of sequences over time. So basically looking after each round of, after each biopanning round, we can see how clusters or individual sequences behave. And by doing this, or a group actually has shown in a paper published 2 years ago that this allows for better prioritization of clones and ultimately leading to a higher number of relevant hits found. And as I already alluded to, due to the higher sensitivity of next generation sequencing, we're actually also able to find sequences that would have otherwise been lost when using conventional sequencing strategies like Sanger sequencing. So here I'm actually showing you a plot from this paper from 2 years ago. So it shows how it shows some data from this recent paper where next generation sequencing was compared to conventional screening methods, in this case Sanger sequencing. And by tracking these enrichments over time, which you can see, basically what they did here was track these clusters of sequences over time. You can see, you can track the enrichments of these clusters, and the authors were able to find not only all sequences that were found with conventional methods, but also they discovered a substantial number of novel antibody hits that were not found using Sanger sequencing. So another interesting example of how RepSeq data can be used is to infer sequence lineage trees that map the evolution of, for example, a B-cell response. So in this particular example, every node here is an antibody sequence, starting from the germline sequence depicted here in red, and further arranged by how the sequences matured or evolved over the course of, for example, the process of somatic hypermutation. So you can imagine that we can use external information obtained in the lab, like affinity measurements, or through a computational analysis of the sequence, and overlay this tree with additional layers of information. So in this particular case, you can think of sequence properties, like, for example, how far the sequence has diverged from the germline, presence of amber codons, or other developmental liabilities, like, for example, glycosylation or deamination sites, these kinds of sequence properties. And what's nice about this kind of visualization is that it allows you to discover close neighbors. So it's depicted here. It allows you to discover close neighbors of sequences that you might have characterized in depth through external assays or external experiments, and find sequences in the neighborhood that might actually have better properties, like better affinity or different developmental liabilities. So as might be clear by now, NGS can generate vast amounts of data. So to extract the most from this kind of data, we need an integrative system which can capture all the various informations from a multitude of samples obtained longitudinally, for example, and also be annotated with other sources of information, like, for example, wet lab experiments or measures that have been computationally inferred. And another benefit would be to have historical data at hand, which would allow researchers to spot historical trends or combine experiments or combine data, from different experiments. For example, to avoid repeating mistakes or to link particular sequence traits to developmental liabilities. In addition, there is, of course, also a huge wealth of information that is now available in public databases, like, for example, Immune Access, or IMGT, or other public sequence databases. These discovered sequences could also be implicated in diseases other than the ones which you might be happening to study. So to allow researchers to perform these kind of analyses and to manage their data in an efficient way, and so to support antibody discovery and development efforts, we developed the IGX Platform, which is basically an end-to-end platform where researchers can store, manage, analyze, visualize all their immune receptor information in one place. So we built the platform in a modular structure. So we have apps that range from processing raw sequence data into annotated sequences, as well as other apps which analyze and visualize those sequences. So all apps are connected to the core of the platform. I think we're missing a little bit of contrast here, but there's some better organization, I think, than you're able to see here. So all apps are connected through the core of the platform, and this is essentially, the core of the platform is essentially the place where all raw data and the processed sequences are stored, and it's essentially the starting point for any analysis. So upstream of the core, we have a number of apps for QC and for clone annotation. IGX Inspect allows you to visualize some quality control plots, and IGX Profile allows you to go from raw FASTQ sequence data into annotated and quantified clones, like, for example, BCRs. And downstream, currently we have IGX Explore, which is an app which allows calculation of various diversity metrics, like Shannon entropy or Simpson dominance, but also visualization of your clonal data, like, for example, visualizing CDR3 length distributions or clonal overlap between multiple samples. So one of the most important features of the platform is the ability to use tags. So it's we basically built the whole system around this concept, a flexible and extensible tagging system. So the system allows users to link their sequencing data, both on the sequence level or on the sample level to any kind of metadata or even experimental data. So tags can be either descriptive, like a study identifier or a disease type or tissue type, but they can also be quantitative, like, for example, annotating affinity data or any kind of quantitative data that you might be generating in the lab. And through this tagging system, you're basically able to organize your repertoire data and also probe it in virtually infinite ways. One other benefit of using this kind of tagging system and also having it integrated in such a profound way is that you can imagine that in the phylogenetic tree analysis that I just showed you, you might use the tag data to overlay information on these kinds of plots. So basically use affinity data and overlay this in, for example, a phylogenetic tree analysis like shown previously. So I hope I've shown you a number of ways in which NGS and also the IGX platform can support the effort in the antibody discovery and development field. Just quickly summarizing, we devised a way, or we developed a platform which allows you to directly link experimental data to antibody sequences using a tag-based system. We support the use of NGS to find candidate enrichments in biopanning rounds. I've shown you the phylogenetic tree analysis that we think is pretty powerful and might help discovering novel antibody sequences with different features, like different developmental liabilities. And finally, having all the information stored in one place will allow you to track trends over time in your in your experimental data, but also probe external databases, which can provide a wealth of information about also your own sequences. So if you'd like to discover how ENPICOM and the IGX platform can improve your antibody discovery workflow, you can visit us at our booth. We're at booth A downstairs. And yeah, thank you for your attention. And if there are any questions, shoot.